Hello, everybody. It's so nice to see such a great crowd of people to come and hear today's wonderful lecture. And also welcome to all of you who are tuning in through the live stream. So, very exciting day for us. As we gather today, we acknowledge that Vassar stands on the homelands of the Monje Lenape, indigenous peoples who have an enduring connection to this place despite being forcibly displaced by European colonization. Monje Lenape peoples continue today as the Stockbridge Muncie community in Wisconsin, the Delaware tribe and the Delaware Nation in Oklahoma, and the Muncie Delaware Nation in Ontario. This acknowledgement, however, is insufficient without our reckoning with the reality that every member of the Vassar community since 1861 has benefited from these Native peoples' displacement. And it is hollow without our efforts to counter the effects of structures that have long enabled and that still perpetuate injustice against Indigenous Americans. To that end, Vassar commits to build and sustain relationships with Native communities, to expand opportunities at Vassar for Native students, as well as Native faculty and other employees, and to collaborate with Native nations to know better the Indigenous peoples, past and present, who care for this land. I'm absolutely thrilled to introduce an individual who is a creative, courageous, and caring scientist-physician. In her lab, Dr. Monje and her team are breaking new ground in neurodevelopment, and at the same time, in her clinical practice, she is a pioneer working to develop treatments for a deadly and aggressive childhood brain cancer. The Richard Lounsbery Award is given in alternate years by the National Academy of Sciences and the French Academy of Sciences to young French and American scientists to recognize extraordinary scientific achievement in biology and medicine. In addition to honoring scientific excellence, the award is intended to stimulate research and encourage reciprocal scientific exchange between the United States and France. Dr. Monje received this honor in 2023, and as part of the award, she chose to give her award lecture here at her alma mater. During her visit, Dr. Monje is sharing her path from college to being a career scientist in her field, and it's just been a wonderful afternoon already for us to meet with her and learn from her experiences. While at Vassar, biology lit a spark for Michelle, particularly courses in microbiology, biochemistry, immunology, organic chemistry, and developmental biology. She also took courses in Romanticism, French, Jewish history, and the Russian novel. She honed excellent writing skills that you see today in clear and interesting prose, in review articles, scientific reports, and public speeches. From the beginning, Dr. Monje gravitated toward the challenges and rewards of research, pursuing independent work in biochemistry with Professor Bill Strauss, including a summer at the Mount Desert Island Biological Lab as part of the Undergraduate Research Summer Institute program, or URSI, here at Vassar. Then she joined my lab, where she developed her own series of studies investigating the potential protective effects of vitamin C and vitamin E on the oxidative damage wrought in rat brain neurons resulting from the loss of oxygen and glucose, or experimental stroke. This collaboration, which lasted till her graduation, shifted the focus of the work that I had been doing and was a fruitful line of inquiry for quite a few years. Here's her first lab notebook, filled with careful notes and observations. And Michelle, I'm giving it to you because one day you may want it as you write your memoir. Her work led to a publication and also propelled her towards her dual pursuit of research and medicine. After Vassar, Dr. Monje earned a PhD in neuroscience and an MD from Stanford University with residencies at Massachusetts General Hospital and Brigham and Women's Hospital, Harvard University Medical School. Dr. Monje is currently Milan Gamir Professor of Pediatric Neuro-Oncology and Professor by courtesy of Neurosurgery, Pediatrics, Pathology, and Psychiatry and Behavioral Sciences, with a primary clinical appointment in the Division of Child Neurology. Her honors and awards are way too numerous, even already, but just in the past 5 years, investigator for the Howard Hughes Medical Institute, a MacArthur Genius Award, the Paul Marks Prize in Cancer Research. She's a member of the National Academy of Medicine and received the Presidential Early Career Award for Science and Engineering, as well as the Jonathan Kraft Prize for Excellence in Cancer Research. One of the world's premier specialists in diffuse intrinsic pontine gliomas and spinal diffuse midline glioma, Dr. Monje and her collaborators have published in Science, Nature, Neuro-Oncology for totaling more than 150 publications, contributing important and paradigm-shifting new understandings of the role played by the microenvironment of glia and neurons during key stages of neural development. The full collaboration between developing neurons and glia, so crucial for the development of functional circuitry, becomes dysfunctional during aggressive cancers that can affect children as young as 6 years old. Her work is carefully revealing these interactions, how they go awry, and developing approaches to intervene that are opening up exciting therapeutic avenues for the treatment of these cancers. So without further ado, Dr. Monje will tell us about her work.
I don't have the words to express what an enormous honor it is to be back here, and what a pleasure it is to kind of come home to Vassar. So thank you so much for hosting this lecture. Broadly speaking, my laboratory tries to understand the molecular language that cells use as they work together to build and to remodel the brain. And I want to tell you one story today in 2 parts about the way in which neurons interact with the glial cells that form the myelin sheath, how we're increasingly understanding that this is an experience and activity-dependent process that tunes neural circuits in ways that are important for a range of brain function, and how these really powerful interactions between neurons and glial cells are hijacked and subverted in the context of glial malignancies. But I like to start every talk with a broad consideration of neural development, and I think the best way to do that is to show you pictures of my children. So this is my daughter Sophie when she was an infant. Sophie was our 4th child, and it occurred to us by the time we had Sophie that we needed to give her some way to keep track of the enormous number of neurodevelopmental to-dos that humans have to accomplish every day in the first year of life. It is really astounding when you consider that humans make something on the order of 500 billion synapses every single day in the first year of life, approximately 14,000 new hippocampal neurons, 127 million new cerebellar granule cell neurons every day in the first year of life on average. And as impressive as that neuronal development is, perhaps the largest task that we have in the postnatal period is to develop the myelin of the central nervous system. Myelination is a chiefly postnatal developmental process. Myelination begins just before birth, around the center of the brain, and then it progresses in an initial massive wave of myelination towards the poles of the brain over the first 1 to 2 years of life. But it doesn't end there. It turns out that developmental myelination is a very protracted process spanning about 30 years of postnatal neurodevelopment. So looking out at all of the bright faces in this crowd, I see many people who are not yet done with developmental myelination. This protracted process of myelin development does follow some pretty predictable chronological and topographical patterns that generally follow the principle that more basic circuitry, such as that underlies sensation and movement, myelinate prior to more complex neural circuitry, such as that's important for higher cognitive function. So, for example, there's a discrete wave of developmental myelination in the ventral brainstem in mid-childhood, around the time that kids exhibit kind of a stepwise improvement in motor function when they begin to be able to, for example, skip with alternating feet or ride a bicycle without training wheels. And then later, in adolescence and young adulthood, around the time we trust people to drive a car, there's a discrete wave of developmental myelination in the neocortex and intercortical association fibers. Now, this process of myelination, the process by which oligodendrocyte precursor cells proliferate, generate new oligodendrocytes that then ensheath the axonal membrane to decrease transverse capacitance and increase the speed of neural impulse conduction, it doesn't end at the end of neural development, but in fact, in some parts of the nervous system, like the neocortex and its projections, myelination continues throughout the lifespan. And we've appreciated this for, at least 100 or more years since these historical lithographs of human postmortem tissue that shows you that there's this accumulation of myelin in the neocortex over the lifespan. More recently, some really beautiful in vivo live imaging work in rodents has shown this continual addition of new oligodendrocytes, new myelin internodes in the neocortex until old age. Now, I would like to argue that this is an inherently fascinating process and set of observations, but it's also really important to understand because glial malignancies of childhood, adolescence, and young adulthood also happen in a very predictable spatiotemporal pattern. I once had a mentor say to me in clinic, "If you tell me the age of a child, I will tell you where their glioma is." And when we take a step back and think about what developmental processes kind of match with the spatiotemporal pattern of these childhood glial malignancies, they fit pretty well with the spatiotemporal pattern of developmental myelination, such that at a time when there is this discrete wave of ventral pontine myelination, that's when one of the very worst human cancers, diffuse intrinsic pontine glioma, tends to occur in mid-childhood, peaking in incidence around age 7. Similarly, when there's that discrete wave of myelination in the neocortex in adolescence, this is when high-grade gliomas of the cerebral hemispheres and the cortex tend to occur. And these findings are concordant with observations from my laboratory and from many others that many of these glial cancers, even those that we have traditionally thought of as quote-unquote astrocytomas, in fact arise from precursor cells in the oligodendroglial lineage, either bona fide oligodendrocyte precursor cells, or OPCs, or just earlier precursors that are committed to differentiate down an oligodendroglial lineage. And it certainly fits with single-cell molecular studies of these tumors that really highlights a prominent and very functionally important subpopulation of OPC-like malignant cells. And so we may learn really important lessons about gliomagenesis by better understanding this process of normal oligodendrogliogenesis. So that asks a very basic question. What does regulate the proliferation and functional differentiation of oligodendroglial lineage cells? Well, one idea that's been in the literature for some time, first introduced in the 1990s when I was a student here at Vassar by Ben Barris when he was a postdoctoral fellow in Martin Raff's lab, the idea that neurons themselves might regulate the extent to which their axons are myelinated, and that this might happen in an experience and activity-dependent manner. Well, this was a very controversial idea for a number of years because there are also clearly activity-independent modes of myelination. If you place an oligodendrocyte in a petri dish with any appropriately sized fiber, formalin-fixed axons, inert nanofibers, they will wrap anything of appropriate diameter. So we know that there are activity-independent modes of myelin development. But the question remained, in some parts of the nervous system, in some circuits, in some neuronal subtypes, might myelin be shaped by activity? Could myelin actually be plastic and adaptable to experience? And I'd like to focus the first part of this talk on this idea of myelin plasticity, of activity-dependent changes in the myelinated infrastructure of the nervous system. and to introduce you to people who have led this work in my laboratory. This is Erin Gibson. I was very lucky to recruit her as my very first postdoctoral fellow. Erin is now leading her own independent laboratory in the Department of Psychiatry at Stanford. Anna Garrity, who did her postdoc with me and is now, I was lucky enough to keep in my lab as a senior staff scientist. Juliette Knowles, who finished her postdoc and is now running her own laboratory in the Department of Neurology. She's also a pediatric epileptologist. And Belgen Yeltsin, who's a brilliant postdoc in the lab on the job market this year. And what Erin, Anna, Juliette, and many others in the laboratory have done is to take techniques in modern neuroscience and leverage them to understand these interactions between neurons and other cell types, like oligodendrocytes. And so, as probably many in the audience know, but I'd like to introduce, this is a technique we've used very extensively called in vivo optogenetics. And what this technique allows is for us to control the activity in targeted populations of cells that have been engineered to express light-sensitive ion channels isolated from microorganisms. So this is channel rhodopsin 2. It's a cation channel that is sensitive to blue light. It's isolated from algae, and if we express it in cortical projection neurons in the motor planning, or M2 cortex, then we can stimulate action potentials in those cortical projection neurons with flashes of blue light. And if we do that at the frequency at which these motor neurons typically fire, which is 20 hertz, here's a mouse in which we're delivering blue light through an optical ferrule, and now that the light is flashing at 20 hertz, it's beginning to execute complex motor behavior. And this simple behavioral readout lets us know that we have successfully recruited activity in the circuit. And then we can ask really straightforward questions about how other cell types respond to this change in cortical projection activity. And what we discovered was that cortical projection neuronal activity results in rapid and robust proliferation of oligodendrocyte precursor cells in a circuit-specific and neuron subtype-specific way. We can fate map those cells over time by administering the thymidine analog, EDU, at the time that we deliver the optogenetic stimulation. And then we find that those proliferating oligodendrocyte precursor cells differentiate into mature oligodendrocytes, and that the myelinated infrastructure of that circuit changes. And it changes in a way that we would predict might alter conduction velocity, circuit dynamics, and perhaps behavior. And indeed, what we found was that there was an improvement in mouse motor behavior that depended upon the generation of new oligodendrocytes and these myelin changes. So, wanting to understand the mechanisms that mediate these neuron-glial interactions underlying myelin plasticity, we hypothesized that activity-regulated neurotrophin signaling might be one important mechanism. And we tested this idea that the activity-regulated secretion of the neurotrophin brain-derived neurotrophic factor, or BDNF, signaling to its receptor TrkB on oligodendrocyte precursor cells may be one key mechanism of this neuron-glial communication. We tested this using two different genetically engineered mouse models. In the first mouse model, made by Mike Greenberg's lab at Boston Children's Hospital, there are normal baseline levels of BDNF, but the mice have been engineered to not be able to increase BDNF expression or secretion through deletion of the Krebs binding site in promoter 4 of the BDNF gene. In a second mouse model, rather than modulating the ligand, we modulated the BDNF receptor just in oligodendrocyte precursor cells and in an inducible way. And we find that in either of these genetically engineered mouse models with impaired activity-regulated BDNF secretion or the ability of the OPC to respond to that ligand, that the activity-regulated response is entirely lost. So in comparison to either BDNF or TrkB wild-type mice, in which neuronal activity, evoked optogenetically, increases OPC proliferation, in the absence of intact BDNF to TrkB signaling, there's no activity-regulated change in OPC proliferation, no new generation of new oligodendrocytes, and no changes in myelin. And so while we think that the mechanism mediating these interactions between neurons and oligodendroglial cells is much more complex than just this one signaling pathway, this gave us a molecular handle to specifically block activity-dependent myelination and begin to ask what role this process may play in a range of brain functions. And so, for example, testing the role of activity-dependent myelination in intact attention and short-term memory function, we tested this in a behavioral task called the novel object recognition test, which is a very straightforward behavioral test. We put a mouse in a chamber with two identical objects, in this case, two identical Lego structures. So it's always fun to walk into the behavior room and see my postdocs playing with Legos. So they, are inherently novelty-seeking. Mice like to interact with new things. They'll spend some time interacting with and exploring these two objects. We then remove the mouse from this chamber and replace one of these objects now with a novel object. Now, a mouse that had paid attention to and can remember what it previously interacted with will spend a little bit more time with the novel object. But what we find is that if we prevent activity-dependent myelination for a few weeks before the testing, either at the end of developmental myelination at P35 in the mouse or later in full adulthood, that this behavioral test fails. The mice don't distinguish between the novel and the familiar object, indicating an impairment in attention and or short-term memory function. And so using similar strategies, now there's been a great deal of work in the field, and we've discovered as a field that modulating neuronal activity, either using optogenetics, chemogenetics, changes in experience like social experience, visual experience, somatosensory experience, all of these activity-regulated changes influence myelination of the relevant circuits and contribute in important ways to a range of brain functions, including attention, memory, learning, and motor function. The adaptive changes that we see in the healthy nervous system take a couple of different forms. I've been talking so far about de novo myelination, about proliferation of a precursor cell, generation of a new oligodendrocyte, and then establishment of a new myelin internode on previously unmyelinated axons or discontinuously myelinated axons. In addition to this de novo myelination, there's also certain circuits that instead exhibit myelin remodeling by existing oligodendrocytes, in which an existing oligodendrocyte changes the geometry of the internode in a way that would subtly influence conduction velocity. Those internodes can become longer or shorter, fatter or thinner. The mechanisms that mediate these changes is an area of intensive investigation, and they generally fall into two categories. As I've told you, there are activity-regulated growth factors that are secreted. There's paracrine factors that can be secreted, like BDNF. Another one that was discovered by Charles French Constant's group is endothelin. But in addition to this activity-regulated growth factor secretion, there's also, fascinatingly, bona fide synapses that form between neurons and oligodendrocyte precursor cells. These were first described by Dwight Burgles about 25 years ago. They take on both glutamatergic and GABAergic flavors. They're very well characterized. These neuron to OPC synapses are present on just about every OPC. But the role that they play in activity-dependent changes in myelin is still really poorly understood. In recent years, we've used additional techniques in modern neuroscience like monosynaptic viral tracing to try to understand the circuit-level inputs into OPCs, to understand essentially who the OPC is listening to. And we find fascinatingly that OPCs are really receiving inputs from neurons throughout a brain-distributed functional circuit. So for example, if we do monosynaptic viral tracing of OPCs in the motor cortex, we see that they are receiving synaptic inputs from the entirety of the motor cortex, as well as the motor nuclei of the thalamus, the contralateral motor cortex, and the ipsilateral somatosensory cortex. So really interestingly, these OPCs are positioned to integrate information from throughout a circuit, but how they do that and how that might contribute to the very precise nature of these activity-regulated changes remains to be understood. What is increasingly clear, though, is that in the healthy nervous system, these adaptive changes in myelin tune neural circuits to promote coordinated circuit function. This is evident both from computational models as well as from experimental work. And one form of this kind of circuit-level coordination is to promote oscillatory synchrony between two nodes in a neural network. So if myelin plasticity plays important roles in healthy attention, learning, and memory, it stands to reason that disruption of this process might contribute in important ways to diseases of impaired cognition. And we found that indeed, impairment of myelin plasticity is contributing importantly, at least in preclinical models, to cancer therapy-related cognitive impairment, to the brain fog that occurs after COVID, and in the learning impairments and disabilities that are evident in children with a neurogenetic syndrome called neurofibromatosis type 1. And I want to dig into a couple of these examples as vignettes to better illustrate this point. As a neuro-oncologist, one of my deep interests is the neurobiology of the cognitive impairment that happens very frequently after cancer therapies. This is a very persistent and troublesome syndrome characterized by impaired attention, the speed of information processing, memory, and executive function. Wanting to understand more about this, we modeled the administration of a chemotherapy that's particularly associated with cognitive impairment called methotrexate. It's an antimetabolite agent. And we gave this in high doses to mice in a way that mimics the clinical regimens. What we found is that methotrexate directly activates the brain's innate myeloid cells called microglia. These reactive microglia then in turn induce neurotoxic astrocyte reactivity, and together these activated reactive microglia and astrocytes disrupt both myelin homeostasis as well as completely prevent myelin plasticity. We know that this immune cell, this microglia, is central to this pathophysiology because if we target it, in this case experimentally using a small molecule CSF1R inhibitor, that restores myelination and it rescues cognition. So we know that this disruption of myelin homeostasis and plasticity is really quite central to the cognitive phenotype that we observe. Now, when the pandemic began, I was really worried that given that this is fundamentally an immune-based mechanism, that we might begin to see the same kind of cognitive impairment syndrome in people surviving COVID, even relatively mild COVID, given how profoundly immunogenic this infection was. And it became evident within a couple of months of the pandemic that indeed people were coming to the neurologist complaining of a syndrome that looked almost identical to cancer therapy-related cognitive impairment. And so I wanted to study this, but we were in deep lockdown, and I reached out to, and also I'm not an immunologist, so there was also that kind of challenge. So I reached out to a real leader in immunology and virology who had recently developed some very faithful mouse models of COVID named Akiko Iwasaki, and asked her if she would collaborate with us to test this hypothesis. Dr. Iwasaki had developed a model of mild respiratory restricted COVID. So these are mice that have been engineered to only be infectable with the virus that causes COVID, SARS-CoV-2, in the respiratory system. It's a pretty mild infection in these mice. They clear the virus in about a week. They don't lose weight or show any impairment in grooming. They really have a very mild clinical course. And yet, when we look at their brains, they have the same pattern of axon-tract microglial reactivity that we see after cancer chemotherapy. And as we would predict, this is associated with a loss of mature oligodendrocytes in the axon tracts and a loss of myelinated axons. When we compare the loss of myelinated axons that we see after this mild respiratory restricted COVID infection, either 1 week after infection or 7 weeks after infection, when we compare that to the degree of myelinated axon loss that we see after methotrexate chemotherapy, it's really quite similar. So very, very similar and parallel pathophysiological mechanisms in these various brain fog syndromes, all involving disruption of myelin homeostasis and plasticity. So I hope I've convinced you that myelin plasticity is important in normal cognitive function, that it is implicated in diseases of impaired cognition. What happens in diseases that are characterized by abnormal levels of activity or abnormal patterns of activity? Might this activity-dependent myelination change circuit dynamics in a way that contributes to other disease processes? Well, we tested this in the context of both generalized epilepsy as well as reward learning associated with opiate addiction. I want to tell you 2 more vignettes about maladaptive myelination, about too much myelin establishment in the context of diseases of elevated activity. Juliette Knowles, when she came to my lab as a pediatric epileptologist, really wanted to understand whether aberrantly increased myelination within a seizure network might promote the hypersynchrony between nodes in the seizure network that engenders seizures. And we hypothesized that, once the seizures began, we might observe an increase in myelination of that circuit. And indeed, in collaboration with John Huguenard's lab, looking at two different genetically engineered one rat model of generalized absence epilepsy, a thalamocortical epilepsy syndrome, we see that there is indeed this increase both in the number of myelinated axons as well as the thickness of the myelin sheath. We see this only after the rodents begin to have seizures. So there's a period of time prior to seizure initiation in which myelin looks completely normal. The myelin changes are specific to the seizure network. When we look outside of the seizure network, there's absolutely normal myelin in these mice and rats. And the myelin changes depend upon seizures. This happens to be a seizure disorder that can be prevented with an anti-seizure medicine called ethosuximide. If we administer that and prevent seizures from ever happening, the myelin changes never occur. And so the question was then, in the natural history of this disease, in which seizures become worse over time. This happens not only in rodents but also in patients. Seizures beget seizures, and over the course of a seizure disorder, the number of seizures per day and the severity of those seizures tends to worsen. We wondered whether this aberrant increase in myelination of the seizure circuit might be contributing importantly to that pathophysiology. And so we tested that by preventing activity-dependent myelination in some of the mice, again by conditionally deleting the TrkB receptor just from oligodendrocyte precursors, and find that indeed when we do intracranial EEG in these mice, when they have a seizure, there is a decrease in the synchrony between two nodes in the seizure network, which we call ictal coherence. We would predict then that the seizures may not worsen over time in the same way without myelin plasticity as they do with intact myelin plasticity. And we found that that was the case. In mice that have intact myelin plasticity, the number of seizures that occur per hour worsens with over time with age. And here's a snapshot EEG. You see in 1 hour how many seizures a mouse with intact myelin plasticity has. If we prevent myelin plasticity genetically, that worsening doesn't occur, and the number of seizures that happen per hour are lessened. Switching gears to a different disease characterized by abnormal patterns of activity, Belg and Janssen in my lab wanted to understand whether myelin plasticity, number one, was evident in other kinds of circuits like dopaminergic circuits, and two, whether it might contribute in some way to drug-seeking behavior within the context of opiate addiction. The reward circuitry includes one very important component that projects from what's called the ventral tegmental area to the nucleus accumbens. And we find that if we optogenetically stimulate the dopaminergic neurons in the ventral tegmental area, that elicits myelin plasticity within the ventral tegmental area of the dopaminergic axons. There's an increase in the generation of new oligodendrocytes and an increase in the number of myelinated dopaminergic axons. We wanted to know if administration of drugs of abuse might elicit the same kind of myelin effect. And so we exposed mice to we administered mice either morphine or cocaine. And we did this in the context of what's called the condition place preference test. So mice in this test have a couple of chambers that they can explore, and they always will receive their drug reward in one of the two chambers. Over time, most mice start spending a lot more time in the chamber where they anticipate a drug reward than in the other chamber where they never get it. We found that both morphine and cocaine elicited OPC proliferation and oligodendrogenesis, and as expected, that mice spent more time in the chamber where they anticipate getting their drug reward. So next we asked, is the myelin plasticity that's occurring required for that drug of abuse related reward learning? So we repeated this test, this condition place preference test, administering morphine to mice in one of the two chambers. But now we've conditionally deleted TrkB from OPCs and prevented myelin plasticity in some of the mice. And we find that mice without intact myelin plasticity don't learn where they're going to receive their drug reward. Now we can begin to understand this lack of drug-seeking behavior, the lack of spending more time in this chamber, when we look at dopamine release into the target of the ventral tegmental area. So again, ventral tegmental area, dopaminergic neurons project to the nucleus accumbens. We can measure dopamine release in nucleus accumbens using a technique called fibrophotometry. And we see that in mice with intact myelin plasticity, when they enter this chamber where they anticipate the drug reward, there's a nice strong peak of dopamine release into the nucleus accumbens. In mice without myelin plasticity, there's no such coordinated release of dopamine. And so it makes a lot of sense that they're not learning where to expect their drug reward. I hope I've convinced you that myelin plasticity is interesting, that it contributes in important ways to a range of brain functions, that is deficient in some diseases, and that it's maladaptively increased in other diseases. But what happens if instead of being a healthy oligodendrocyte precursor cell, this oligodendrocyte precursor cell has an oncogenic mutation or set of mutations? Could these really powerful interactions between neurons and oligodendrocyte precursor cells be hijacked and subverted in the context of glial malignancies? So I'd like to spend the second half of the talk on this idea of malignant myelin plasticity, of activity-dependent glioma growth. And to introduce you to the people in the lab who've led this work, this is Hamsa Venkatesh. She was an absolutely brilliant cancer biology PhD student in my lab, stayed with me for her postdoc, and is now running her own independent laboratory at Harvard Medical School. Ewan Pan, similarly brilliant, now running her own laboratory at MD Anderson. Katie Taylor, who I am proud to say just a couple of weeks ago accepted a faculty job at Memorial Sloan Kettering and will be beginning her lab in January of the coming year. And Tara Barron, who is a fantastic postdoc on the job market this year. Now, I'd like to point out that I do have men in my lab. Haven't shown them to date, and I'm going to show you the work of these incredible 4 women, but just to highlight other people in the lab with more gender diversity, we do do have some really, really wonderful cancer neuroscientists in the lab that have a Y chromosome also. So these are the cancers of childhood that I spend all of my time thinking about, that my laboratory studies. These are the glial malignancies of childhood. And as I mentioned earlier, they happen in particular parts of the nervous system at particular ages, which really speaks to this collection of cancers being a disease of dysregulated neural development and plasticity. And just to introduce you to the tumors that I'm going to speak about, this is an optic pathway glioma. This is a low-grade glioma type. It often happens in association with the tumor predisposition syndrome called neurofibromatosis type 1. It can also happen spontaneously. It tends to happen in very young children, and it's low grade, so it's usually survivable, but it can cause some profound disability and often vision loss. In contrast to the low-grade nature of optic pathway gliomas, this is one of the most aggressive cancers imaginable. This is diffuse intrinsic pontine glioma. It tends to happen in mid-childhood, peaking in incidence around 6 to 7. A molecularly related disease that happens in slightly older children is diffuse midline glioma of the thalamus. And then in adolescents and young adults, high-grade gliomas tend to be located in the cerebral hemispheres. This is a cancer that looks very similar to its adult counterpart, adult glioblastoma. And the idea that neurons might be playing a role in the biology of these cancers is actually something that I think neuropathologists maybe have appreciated for almost a century. There was a very prescient and brilliant neuropathologist named H.J. Shear who described what he named at the time the secondary structures of Shear and what we call perineuronal satelitosis, the propensity of these malignant glioma cells to cluster in very tight microanatomical association around mature neurons in the tumor microenvironment and then to invade along axon tracts. The idea that neurons might be important and might be important in an activity-dependent way in the tumor biology is something that I thought a lot about as a trainee. I remember being a neuro-oncology fellow and looking at under the microscope at some of my patients' tumors and just wondering, why are they clustering around the neurons? I want to point out that this particular H&E image comes from a 5-year-old patient that I cared for as a pediatric neuro-oncology fellow, and it was this child's early postmortem tissue donation that enabled us to generate the very first cell culture of DIPG, the very first xenograft mouse model, and it's through similar such incredibly selfless donations of tumor tissue from our patients and their families that we're able to do all of the work that we do. So I just want to acknowledge that incredibly important contribution from patients. So to ask whether neuronal activity might be similarly influencing glioma proliferation as it does healthy glial proliferation, we did that exact same optogenetic paradigm, optogenetically stimulating cortical projection neurons, but this time in the context of a diffusely infiltrating cortical glioblastoma isolated from an adolescent patient of mine. And what we found was that indeed neuronal activity drives the proliferation of the malignant glioma cells just like it does their normal counterparts, and that this results in an overall increase in tumor burden, specifically within the stimulated circuit. This was the first demonstration that brain activity can in fact influence brain cancer growth. More recently, and in collaboration with David Gutman's lab, we tested whether neuronal activity was important in low-grade gliomas in the optic nerve in a genetically engineered mouse model that the Gutman lab has developed that's really faithfully recapitulates these NF1-associated optic pathway gliomas. And we found that if we optogenetically stimulate the optic nerve, these retinal ganglion cell axons in the optic nerve, that the tumors that are evident later on, so we do the stimulation beginning at 6 weeks, these tumors typically initiate around 9 weeks. If we look at 16 weeks of age, the tumors that are there are much larger than in littermate controls, brothers and sister mice, that were identically manipulated, but in whom we just simply did not turn on the blue light. Now, this optic pathway glioma model was a really exciting opportunity to ask questions not only about tumor growth, but also about tumor initiation, because we know exactly where and when these tumors are going to occur, and we don't even need optogenetics. These are tumors that happen in the visual pathway. We can just change visual experience to change the activity in that circuit. And so I thought it'd be really cute if we got little light-blocking glasses for the mice, but you and Pan convinced me that that would be a disastrous idea. So instead, we just put the mice into complete darkness at various points around the time of tumor initiation. So these tumors begin at 9 weeks of age. If we dark rear the mice, beginning at 9 weeks of age or even just after, at 12 weeks of age, and then examine the optic nerves at 16 weeks of age, we find that far fewer and much smaller tumors form compared to littermate controls that were simply raised with normal visual experience. And if instead we put mice into darkness prior to tumor initiation, beginning at 6 weeks of age, absolutely no tumors form, even if we replace mice into normal visual experience conditions after what appears to be a period of critical susceptibility to tumorigenesis, and despite the very strong genetic predisposition to tumors in this mouse model. So there are some powerful mechanisms at play communicating between neurons and glioma cells. And we wanted to understand what those were. And we first hypothesized that there may be activity-regulated growth factors that are secreted, activity-regulated paracrine factors, as we had found was the case with normal OPCs. So we tested that idea using a pretty simple experimental setup. We took healthy mouse cortical or retinal explants, and then we collected the secreted factors into the conditioned medium when the neurons in the explants were at varying levels of neuronal activity. So they were either spontaneously active, optogenetically stimulated, or silenced with the voltage-gated sodium channel blocker tetrodotoxin. And what we found is that when we then took those secreted factors in the conditioned medium and placed them onto cultures of glioma cells, that there was an activity dose-dependent increase in glioma cell proliferation, blocked when the activity of those slices was prevented with tetrodotoxin. Now, I've just been telling you that there are a number of different clinically and molecularly distinct glioma types, but we find that the response to these activity-regulated paracrine factors is conserved across multiple different forms of both pediatric and adult glioma, including adult glioblastoma, adult anaplastic oligodendroglioma, pediatric pontine diffuse midline glioma, or DIPG, and pediatric hemispheric gliomas. So the next question was, well, what are the factors that are in the conditioned medium? And so through a couple of years of work that I'm going to summarize with a single text box, as we do, we found that there were two key paracrine factors. Not surprisingly, BDNF, just as it plays a key mitogenic role in normal myelin plasticity, is also playing a role in promoting malignant glioma proliferation. But really unexpectedly, we found a shed form of a synaptic adhesion molecule called neuroligin-3. Now, neuroligin-3, first discovered by Tom Sudoff, is a really important postsynaptic adhesion molecule that modulates synaptic strength and synaptic function. There's a point mutant form of neuroligin-3 that contributes to an autism spectrum disorder. It's been a very well-studied protein, but it was not known to be a mitogen in any context, and actually it wasn't even known to be cleaved and secreted. But what we find is that neuroligin-3 is cleaved at the membrane, which releases this large N-terminal ectodomain. This occurs in a strictly activity-dependent manner through the enzymatic activity of the metalloprotease ADAM10. So the next question that we wanted to ask was, what cell types are shedding neuroligin-3? When we look at gene expression databases, we see that there's very high neuroligin-3 expression as expected in neurons, but also in oligodendrocyte precursor cells, which again are a postsynaptic cell type. So we did some genetic mouse modeling experiments in which we conditionally deleted neuroligin-3 either from subtypes of neurons or from OPCs, and we find that the chief cellular source of shed neuroligin-3 is the OPC, asking some very interesting questions about the role of neuroligin-3 in myelin biology, but also placing OPCs in the tumor microenvironment together with neurons. So the next question we asked was, does this matter? There are many cell intrinsic mechanisms by which tumor cells proliferate and grow, and a number of microenvironmental mechanisms. So we wanted to understand the relative importance of neuroligin-3. And to do that, we simply xenografted patient-derived brain cancer cells into the environment of either the neuroligin-3 wild type or neuroligin-3 knockout brain. And what we found really surprised us. These tumors didn't just slow in their growth, they utterly stagnated in the absence of neuroligin-3 from the tumor microenvironment. Here you see these GFP-labeled tumor cells that we xenografted to cortex. They are proliferating, they're expanding, they're invading across the corpus callosum. They're acting as their human counterpart did. This is a pediatric glioblastoma. In the absence, though, of neuroligin-3 from the brain microenvironment, here are the cells that we xenografted 6 months prior. They persist, but they don't expand. And we can monitor this in real time using in vivo bioluminescent imaging, in which you can see that there is just this stagnation of tumor growth in pediatric glioblastoma, in adult glioblastoma, in diffuse intrinsic pontine glioma, either in the cortex or in the brainstem. So we next wondered about this optic pathway glioma model. Going back to the optic pathway glioma model, we wondered whether neuroligin-3 might be playing a key role there too. And we wondered that not only because it was so important for high-grade glioma growth and progression, but because we noticed that in the optic nerves of both mice and humans with NF1, that there was an aberrant increase in neuroligin-3 shedding. And we wondered whether that represented part of the predisposition to tumors in that particular structure. So to test this, we simply crossed this optic pathway glioma mouse model with the neuroligin-3 knockout mouse and found that this largely phenocopied the results of dark rearing. Far fewer and much smaller tumors form in the absence of neuroligin-3, even with normal visual experience. So what is going on with this interesting synaptic adhesion molecule? Well, we find that when neuroligin-3 binds to the tumor cell, that it recruits numerous oncogenic signaling pathways. There's early upstream stimulation of focal adhesion kinase, downstream SARC, RAS, and PI3 kinase pathways, and that helps us understand the sufficiency of neuroligin-3 in promoting growth, but it really doesn't at all explain the surprising dependency. And so we dug deeper and we looked at the gene expression changes attributable to neuroligin-3 and found that, fascinatingly, neuroligin-3 is regulating the expression of a range of different families of genes, including a number of processes that are very important, we now know, in the way that neurons and brain cancer cells interact. But the one that caught our attention most prominently were these synapse-associated genes. Neuroligin-3 is regulating, number one, a feed-forward effect on its own expression in the glioma cell, together with upregulation of the BDNF receptor TREC-B, encoded by the gene NTRK2. But in addition to that, genes encoding a number of different glutamate receptor subunits and other synapse-associated proteins. So that raised in my mind what seemed at the time like a really crazy question. Could there be synapses between neurons and malignant glioma cells the way there are synapses between neurons and OPCs? Well, when we look by immunoelectron microscopy, in which we can unambiguously identify the tumor cells through immunogold labeling, there are very clear synaptic structures with these presynaptic neurons containing synaptic vesicles, a clear synaptic cleft, and a postsynaptic density on the cancer cell. testing the idea that maybe neuroligin 3 is functioning in its most fundamental way by regulating the establishment of these neuron to glioma synapses, we see that far fewer of these neuron to glioma synapses occur in the absence of neuroligin 3 from the microenvironment. But the question remained, is this just a shadow of the cell type from which we believe these tumors arise, or might these neuron to cancer synapses actually be electrophysiologically functional? Well, we tested this using whole cell patch clamp electrophysiology, xenografting these brain tumor cells into a really experimentally tractable circuit in the hippocampus. And in this way, we can patch clamp cells that are in CA1 while stimulating the axonal afferents into CA1. And what we find is that in the cancer cells, there are very clear excitatory postsynaptic currents. These depend upon action potentials. They're blocked by tetrodotoxin. They exhibit multiple electrophysiological characteristics of bona fide synapses, including paired pulse facilitation and single vesicle events called mini EPSCs. And this first kind of synapse that we identified are mediated by calcium permeable AMPA receptors. Now, I have to share that, this really seemed wild when we first found them, and I was very, very pleased when I went on a academic trip to Heidelberg and sat down in Frank Winkler's office and discovered that he had just discovered the exact same thing in glioblastoma. So it's really wonderful when you find something crazy to know that somebody else has found the same thing. So in the healthy nervous system, AMPA-mediated synapses exhibit mechanisms of adaptive plasticity. They change in strength in an activity and experience-dependent way. And that's very important for learning and for memory. We wondered whether the malignant glioma cells were also exhibiting synaptic plasticity. And looking at their gene expression profile, the mechanism of synaptic plasticity we thought most likely would be mediated by a BDNF to TrkB signaling. And indeed, what we find is that if we're patch clamped onto those tumor cells and we perfuse BDNF over those xenografted slices, that the inward current in the malignant cell is much larger, shown here in this blue trace. That depends upon TrkB expression in the cancer cell. If we CRISPR delete the gene that encodes TrkB, the BDNF receptor, then there's no effective BDNF on the magnitude, on the amplitude of these synaptic currents. And what we discovered is happening is that the cancer cells are hijacking a classical mechanism of postsynaptic plasticity whereby BDNF to TrkB signaling through CAMK2 increases the trafficking of AMPA receptors to the postsynaptic membrane, thereby increasing the strength of these malignant synapses. What about other types of synapses? What about other types of neurons? Well, we next looked at GABAergic synapses and GABAergic interneurons, and we find, while we found those AMPA-dependent synapses in every type of glioma that we examined, in the case of the GABAergic synapses, we only find these in these pediatric diffuse midline gliomas like DIPG. We see very clear evidence of GABAergic synapses by immuno-EM. In this case, where the immunogold is labeling a subunit of the GABA receptor in the tumor cell. We fused GFP to a subunit of the GABA-A receptor and then expressed that in tumor cells, co-cultured them with neurons, and I think you can really appreciate that these blue tumor cells expressing GFP, sorry, that's a little jumpy, this GFP-fused GABA receptor are co-localizing very clearly with presynaptic synapsin, shown in white, on these red axons. We also see electrophysiological evidence of these GABAergic synapses. These are sensitive to GABA-A blockers like picrotoxin and bicuculline. Now, we think about GABA as a hyperpolarizing inhibitory neurotransmitter, and it certainly is for mature neurons, but actually in neural stem and precursor cells, including in oligodendrocyte precursor cells, GABA is instead depolarizing. And whether GABA acts as an excitatory or inhibitory neurotransmitter really just depends upon the chloride concentration in the cell. Well, we find that the chloride concentration is very high in these pediatric diffuse midline glioma cells, and that GABA is depolarizing. We also find that the GABAergic current in the cancer cell is augmented with commonly used positive allosteric modulators of GABA signaling like benzodiazepines. So this would suggest that, like glutamatergic neurons promote glioma growth, that GABAergic interneurons might also promote glioma growth. And we tested that, again, using optogenetics, in which we optogenetically stimulate GABAergic interneurons and find that that increases the rate of proliferation of the tumor cells in diffuse midline gliomas. Now, we give benzodiazepines, which augment GABAergic signaling, to these patients not infrequently for a range of different reasons, for nausea, for sedation, and for other reasons. And we wanted to understand if actually in this particular tumor context that might instead promote growth. And we find that, somewhat alarmingly, indeed, when we administer the benzodiazepine lorazepam or Ativan to mice bearing diffuse intrinsic pontine glioma, that increases the rate of tumor cell proliferation in a dose-dependent way, resulting in an overall increase in tumor growth and decreased survival of the mice. So just as it's very important to identify neurophysiological medicines we might be able to repurpose to disrupt these neuron-glioma interactions, we also need to understand which neurophysiological medicines might not be indicated in particular disease contexts. So what I've been telling you is that these tumors are integrating into the circuits that they invade. They're doing this through bona fide neuron-to-glioma synapses that are then elaborated and amplified through mechanisms of adaptive plasticity. They also are purely electrically integrating through potassium-evoked currents that are amplified in a gap junction-coupled tumor-to-tumor network. That's a lot of different ways that a tumor cell is developing to undergo membrane depolarization, which is a very metabolically expensive thing to do. So there must be some advantage to the cancer to undergo membrane depolarization as rapidly and frequently as it's doing. And you know, that makes some sense. We know that there are depolarizing waves of membrane depolarization and consequent calcium transients that happen in the developing germinal zone of the brain during corticogenesis. And that's actually really important for regulating cortical precursor and stem cell proliferation and differentiation. So it may be that there are some voltage-regulated signaling cascades that are being hijacked by the cancer cells in this context. So we wanted to test that idea, and we again used optogenetics, but this time expressing these light-sensitive ion channels not in the neurons, but rather in the tumor cells themselves. And in that way, we can use blue light to depolarize the cancer cells. If we xenograph those cells to the mouse brain and then optogenetically depolarize the tumor, that does in fact promote its proliferation and growth. Now, the tumor cells are not just undergoing membrane depolarization, but they're quite actively recruiting mechanisms to increase the strength of the current, to increase the size of the depolarization. So we tested the idea, again using optogenetics, that larger depolarizations may differentially promote tumor cell proliferation and growth. And indeed, using optogenetics to either invoke a medium-sized depolarizing current or a larger depolarizing current, we find that there is a depolarization dose-dependent effect on glioma cell proliferation. We can visualize the electrical activity in these cancers using genetically encoded calcium indicators, and I think this particular calcium imaging movie of a diffuse intrinsic pontine glioma underscores what for me has been a really startling new understanding that this cancer is an electrically active tissue. And that is not how we have been approaching either understanding it or treating it. And so as a field, we really now need to understand the mechanisms of malignant circuit assembly, plasticity, and evolution over the disease course, to figure out the granular details of these voltage-sensitive mechanisms of proliferation. And I'd like to propose that in doing so, we're going to glean really important insights for normal development and plasticity viewed through the magnifying lens of these glial cancers. Already, in the beginning of our understanding of this, we're seeing some potential opportunities for therapy. And there really does seem to be great therapeutic potential of disrupting these neuron-glioma interactions. Any one of these interactions that we have disrupted slows growth and extends survival in mouse models. And one example of that is disrupting the AMPA-dependent signaling. Now, we can do that with some AMPA-targeting anti-seizure medicines. We can also do it by expressing dominant negative versions of the AMPA receptor subunit GluA2 in the tumor cells. And you can see here this start growth inhibitory effect of preventing the neuron to glioma synaptic communication. What's emerging through now what has been quite an exciting kind of explosion of work in this field is that there is a bidirectional communication between the neurons and the tumor cell network that forms. And so just as neurons are promoting glioma proliferation and growth through paracrine signals, through bona fide neuron to glioma synapses, the tumor cells increase the excitability of neurons. This contributes to tumor-associated seizures, and also, alarmingly, functionally remodels neural circuits. This has been demonstrated not only in rat models and mouse models, but also in humans. As we begin to understand these interactions, a number of potential targets for therapy are emerging. We may be able to target neuroligin-3 cleavage and or binding, AMPA-dependent signaling, glutamate reuptake, neurotrophin signaling, synaptogenic factors, potassium channels, gap junctions, and in the case of diffuse midline gliomas like DIPG, this GABAergic signaling. I want to end with the further implication for this emerging understanding of the neuroscience of these cancers for immuno-oncology. While this is the topic of an entirely different talk, and I'm only going to present one slide of it today, we have been leading some of our discoveries in the laboratory in some very promising immunotherapy CAR T-cell mediated trials at Stanford. And I want to show you this one example of a child with a diffuse intrinsic pontine glioma that has had a complete response, durable for over 3 years now, which is amazing. But in the majority of our patients, the response to immunotherapy is only partial and insufficiently durable. And part of why that's true is that these tumors just grow so quickly, it is difficult for CAR T cells to outpace the tumor growth. And so by leveraging some of these insights in cancer neuroscience, might we be able to slow the tumor enough that the CAR T cells can outpace the tumor growth? Furthermore, it turns out that there is extensive crosstalk between neurons, immune cells, and cancer cells. And as we understand those neuronal mechanisms that may be modulating the tumor immune microenvironment, we may be able to further optimize and augment immunotherapy in the context of the nervous system. I think that the mechanistic parallels evident between normal neuron-glial and malignant neuron-glioma interactions really underscores the extent to which these cancers are just hijacking mechanisms of neural development and plasticity and demand that we begin to approach these cancers from a neuroscience perspective. There are many people to thank in my laboratory, past and present. This has really all been very much a team effort. Collaborators, funding sources, the patients and families whose donation of tumor tissue enables all of the work that we do. And I want to especially thank the Lounsbery family and the National Academy of Sciences Award Committee for enabling me to give this lecture here in my alma mater. And I have to conclude with a big thank you to Vassar. It's been 30 years since I had the privilege of beginning my education here, and it is so clear to me that I, have used what I've learned in my education here throughout the last 30 years. And I am deeply grateful to my professors in science departments who gave me the foundation for, launching my scientific career. I am equally grateful to my professors in humanities departments who taught me really how to critically think and how to creatively think. And I especially need to thank Kate Sussman, who is truly the reason that I am in science. Thank you, Kate. So I know that I went a little bit long, but I think we hopefully have a couple of minutes for questions. I'm certainly not going anywhere, so we can talk afterwards. But if you have a question, please come up to the microphone here in front. And thank you all so much for your attention. Alternatively, I think there's some refreshments in the back.
So when you were talking about the mice and their conditioned place preference,
Yeah.
It was interesting to me that the ones that had, I think, was it the track B knockout, actually seemed to have an exacerbated preference, but going in opposite ways rather than kind of a flat preference. Do you have any thoughts about that? Or?
There was no significant difference between the time they spent in either chamber. Yeah.
So what, was that averaged, or was that like on an individual basis?
Both. Okay. Both the average of the groups as well as on an individual basis. But good question.
Hi. Thank you so much. That was lovely. I was wondering about what do you think about the communication in between glial cells? You talked a lot about the neuron interactions, and I was wondering, especially since we're starting to see these networks going across the brain.
Yeah.
So.
One wonderful question, one of my favorite topics. There is really, I think about every neural cell type sitting at a dinner table having a conversation with each other, and there's critically important glial interactions. Astrocytes form a gap junction mediated network throughout the brain, and that contributes in very important ways. We're only beginning to understand brain function. Microglia and astrocytes clearly regulate each other and communicate and regulate with neurons. There's conversations between every individual relationship imaginable. So they're all very fascinating, but we'd be here for 3 hours if I tried to talk about them all.
I'm actually very interested in the effect of COVID-19 on the brain and glial cells and in general, like how it damages. Where can I find more information from reliable sources on that?
So I tried to summarize everything that was known at the time in a review article that I published about 1.5 years ago in the journal Neuron. So I think it's called The Neurobiology of COVID. No problem.
Hi, thank you so much for your speech. This is like very interesting, and I really enjoy it. And I'm curious about for neurolegend 3, if you inhibit it for the patient's mind, would there have like other side effects on their brain activity?
Yeah. So one really, I think, convenient thing is that in neurolegend 3 knockout mice, neurolegend 1 there's a compensatory increase in neurolegend 1 expression, and the mice are actually very close to normal. They really function quite well. Targeting neurolegend 3 specifically is something we've been working to do. ADAM10 inhibition is one strategy, but ADAM10 is an enzyme that mediates many different processes, and so certainly has a number of side effects when inhibited pharmacologically. We've just identified the binding partner for neurolegend 3, and that may be more specific. It's not entirely specific, but we think it may be better tolerated. And then strategies like antisense oligonucleotides to decrease neurolegend 3 expression in the tumor microenvironment is kind of a third way that one might target that really important interaction.
Can I also have another question as a
Sure.
freshman student? I'm very curious, how did you find your specific field that you want to study? Because now I wanted to study in biology, but I don't know which direction should I choose.
I've always been one to sort of follow my passions and my interests, and being a clinician scientist, that's often inspired by my patients. And so when I first took care of a child with DIPG, I realized I needed to study that disease. I couldn't turn away from it. There was something about neurodevelopment that was going wrong. I started by trying to figure out what it was, and when I realized that there was a link to myelination, I started to study myelin.
Thank you so much.
Yeah.
I also want to say thank you. I sort of came to this talk on a whim, and I was hooked within 5 minutes. And something that really fascinated me was the discussion on how gliomas form synapses between like a normal glial cell would. Because I actually previously, in high school, part of what got me to Vassar was I did research with neurexins, and seeing neurolegend 3 be I mean, I did it with nematodes, so it's a little different, but
But surprisingly the same. So
Yeah.
just for the rest of the audience, neurexins are the canonical binding partner of neuroligands. And
so
Yeah.
the question that immediately came to my mind is, have you? I mean, I think you just mentioned it in that last question a little bit, but have you been looking at how the synaptogenesis occurs between gliomas?
yes,
and
we have in quite a lot of detail. And interestingly, there's a repurposing and the cancer really doesn't invent anything new. It just repurposes other existing processes. And, the same synaptogenic factors, thrombospondins, glypicans that contribute to normal synaptogenesis are important in this malignant synaptogenesis. But interestingly, we kind of thought, well, nerexin must be the binding partner for neuroligin, but it's not. It's not. Stay tuned. Preprint coming.
Hi, thank you so much for this talk. I thought it was really inspiring, especially coming from somebody who's going into the medical field. It's just wonderful to see like the interactions with neuroscience. I worked in a rheumatology department for a little while, and your interactions that you were describing between methotrexate and white matter microglia kind of fascinated me. Mm-hmm. I know that methotrexate is used in lower doses to treat certain rheumatologic conditions. Yeah. So I'm curious if you explored any like dose of methotrexate and like different dosages, and to see like at what threshold the white matter tracts were affected.
This is such a good question. Methotrexate is commonly given for rheumatologic disease at much lower doses than we administer for cancer. We have only tested high-dose methotrexate. It is on my list of things to do a dose-response curve, but we haven't gotten there yet.
Interesting.
Yeah.
Thank you so much.
Absolutely. All right. Thank you for all the wonderful questions and for your attention.